Well, good morning. I'm Chris Fanta. It's my pleasure to welcome you to Asthma Grand Rounds this morning. And we have a wonderful presentation awaiting, but because this is the last Asthma Grand Rounds presentation of the season, I just wanted to take 1 minute to introduce next year's program. We have an exciting lineup, a series of talks ahead that you can see, including one on racial inequities and healthcare disparities and their impact on asthma, an in-depth dive into the biology of resident immune memory in the airways and asthma, a panel discussion detailing thoughts about the use of biologics in the treatment of severe asthma. Then we'll talk about air pollution and asthma by experts in this area. And finally, an exciting pro-con debate about a hot topic in mild asthma, the use of rapid-acting, long-acting beta agonists with an inhaled steroid to replace the use of albuterol in mild and intermittent asthma. And so I hope you'll join us for what I think is now the 30th season of Asthma Grand Rounds coming. Let me remind you that today, if you do have questions, we won't have a chat. Sorry about this error on the slide. All of your questions will need to be texted, and they're texted to this number, so please jot it down, 617-513-6043, and then at the end of the talk, we'll submit your questions to the speaker. And then if you're interested in CME Credit for participation, just email me at cfanta@partners.org, your name and degree and your email address I'll have, and we will submit to Harvard Medical School for your CME credit. And I hope you don't need this, but if you did have any technical problems this morning, here's a rapid response email at bwhwebcasttroubleshooting@partners.org to help with any problems that you might have. So it's a great pleasure to introduce Dr. Sunita Sharma, our speaker today. We welcome her back to Brigham Women's Hospital, where she was a much-beloved faculty member in the Division of Pulmonary and Critical Care Medicine, now Associate Professor of Medicine at University of Colorado Anschutz, where she is in the Division of Pulmonary Sciences and Critical Care. She is a pulmonologist, an intensivist, epidemiologist, geneticist, asthmologist, and just all-around wonderful person. And we're delighted to have her here to talk to us today about intrauterine exposures and the developmental origin of asthma. It's Dr. Sunita Sharma.
Thank you, Chris, for that really warm introduction and for the really amazing opportunity to be able to speak today at today's Partners Asthma Center Grand Rounds. Thank you for this opportunity. It is a humbling and incredible pleasure to be back in the institution that taught me how to be a pulmonary and critical care physician, where, as everybody knows, after this challenging year, it's actually come in quite handy. But in addition to being the place that taught me the importance of scientific inquiry and really gave me the foundation to kind of pursue a career as a clinician scientist. And finally, where I was exposed to incredible role models like the incomparable Chris Fanta, who, if I could be 1/1,000,000th of the clinician, of the educator, of the patient advocate, and just the all-around amazing human being that you are, I would be a success in my career. So thank you, Chris, for the opportunity. I really appreciate it. Today, I'm going to go over some of the work that we've been doing over the last several years, looking at human lung development and how it impacts the developmental origin of asthma. I have no conflicts of interest. So it comes as no surprise to this audience that asthma is one of the most common chronic respiratory diseases worldwide. It's a heterogeneous disease that's characterized by chronic airway inflammation, and it's defined by respiratory symptoms. These are things like wheeze, shortness of breath, chest tightness, and cough that vary over time and in intensity. It's associated with variable airflow obstruction and is at least partially reversible with bronchodilator use. What we've come to recognize when we look at asthmatics is there are potentials for lung decrements over time in patients that have mild to moderate persistent asthma. And this concept is really nicely illustrated by this work from Mike McGahey and colleagues from the Channing Division of Network Medicine back in the New England Journal in 2016. And this is data from the Childhood Asthma Management Program where children with mild to moderate asthma were followed with serial measurements of lung function over time. And what you will see is that there are several patterns or trajectories of lung function that exist in asthmatics. As we know, normal lung function will increase up until the age of 20 when you reach a plateau phase. And then after the plateau phase, we all lose a little bit of lung function as we age. For many individuals, even with mild to moderate persistent asthma, they will get the normal lung growth and will have overall normal lung decline over time. For some individuals, they'll have normal lung growth, but will have early decline in their lung function. And for other individuals, you have stunted lung growth and reduced decline, as well as a pattern that's the most severe, that is reduced growth overall and an early decline. What's notable about these additional patterns is the fact that if you followed these people over time, they developed chronic obstructive pulmonary disease and some level of fixed airflow obstruction. What's really important is that these trajectories appear to be set really early in life, suggesting that early life events, including intrauterine exposures and that can impact lung development, have major implications in terms of lung function development over time. While we recognize that there are a variety of factors that impact lung growth, we have to recognize the influence of both prenatal and postnatal exposures. So some of the prenatal influences that can occur are intrauterine environmental perturbations, things like intrauterine smoke exposure. In addition, maternal health has major implications in terms of the development of the fetal lung. Furthermore, if there are treatment for underlying conditions that exist during pregnancy, things like for infection, for example, so treatment with antibiotics in that context, or the use of corticosteroids for lung maturation, all have major implications in terms of fetal lung development. And notably, in the context of the global pandemic, what we've seen is that there's been increased numbers of individuals being ventilated with mechanical ventilation during pregnancy. And so all of these factors have major influence on prenatal lung growth, but also have influence in terms of postnatal lung function. Furthermore, there are a variety of childhood factors, including nutrition and infection. And as evidenced by much of the work done at this institution, environmental exposures, including both home exposures and at school exposures, can have major impacts in terms of lung development overall. Notably, all of these factors, in addition to genetic predisposition, can actually influence long-term outcomes for respiratory disease. This includes susceptibility to diseases like asthma. Well, there's a preponderance of epidemiologic data suggesting that environmental perturbations that exist in the intrauterine context can actually influence asthma susceptibility postnatally. The biologic mechanisms that really underlie these associations are really not well understood. And so to try and get to the developmental origin of asthma, we use genomic applications to identify the normal patterns of gene expression that exist during human lung development. So the vast majority of what we know about human lung development is really based on human lung histology and animal models. And what we know is that there are 5 histologic stages of lung development that exist. Notably, the lung bud differentiation starts in the embryonic time period when you get differentiation of the anterior foregut. And then you get airway development in the pseudoglandular and canalicular stage. But as we've already discussed, ongoing airway growth occurs into the postnatal period up until that plateau period around 20 years of age. The respiratory gas exchange it develops in the context of both the saccular and alveolar stages of lung development. For the purposes of today's talk, we're really going to focus on this time period of the pseudoglandular stage of lung development. The importance of this time period is that this is when branching morphogenesis or airway development occurs. So if you're interested in looking at the kind of fetal origins of airways diseases like asthma, it's important to see what's happening during normal development and understand what's happening in normal development during that time period, because that's how we can learn from these environmental perturbations how it impacts this time frame. If we look at the pseudoglandular and canalicular stages of lung development, the things that we can really identify based on histology is that there are a variety of different cells that are actually undergoing differentiation. You get epithelial cell differentiation into both columnar and cuboidal epithelial cells. You get airway smooth muscle differentiation. And finally, this is the first time period in which you can actually see the presence of ciliated cells in the proximal airway. And so today we're going to focus on these really two early time points of airway development. And as proof of concept, to look at human lung development, we did genome-wide gene expression profiling in human fetal lung tissue samples from the pseudoglandular and canalicular period. Now, this was in collaboration with a group at the Children's Mercy Hospital in Kansas City who had access to an NICHD-supported fetal tissue repository. We did genome-wide gene expression profiling in these early airway developmental time points. Now, in early work done by Alva and Co. at the Channing, we looked at kind of variability in gene expression in early lung development. And so this plot actually shows principal components analysis during human fetal lung development. And principal components analysis very simplistically looks at variation in gene expression. And what you can see here is that principal component 1, which is found along the x-axis, explains about 45% of the gene expression variation during these early human fetal lung time points. What you can see is that each of these subjects is plotted over time, and that initial phase of gene expression is really captured by estimated gestational age. The reason I show this image is because it's important to recognize that there's a single molecular transition that happens here at day 117 that very nicely characterizes the change in histologic stage between the pseudoglandular and the canalicular time periods. What's notable is that there's an additional transition that occurs here at day 97 of gestational age that's really independent of changes in histologic stage. And what this data tells us is that gene expression can identify some additional novel biology that's important in the context of lung development that's independent of histologic stage alone, and showing the promise of genomic applications in terms of understanding lung development overall. We went on to look at genes that were differentially expressed between the pseudoglandular and canalicular periods. Now, these genes are important because they're dramatically changing in the context of these early lung developmental timeframes. And what we found was that there were about 1,700 genes that were differentially expressed between the pseudoglandular and canalicular stages of development. And these included a variety of really important lung development genes, including the surfactant proteins, as well as the Wnt signaling genes. And for a proof of concept idea, we took genes in these Wnt signaling genes, which we know are influential in terms of lung organogenesis, and we looked to see, are these genes that we now show are important in the context of lung development, do they harbor polymorphisms that impact lung function? And the way we looked at this is we looked at genetic variants in these Wnt signaling genes and looked to see if they were associated with lung function in two well-known asthma cohorts. We first look at genetic variants in these Wnt signaling genes in the Childhood Asthma Management Program, and then replicated these associations in a second cohort of the Genetics of Asthma in Costa Rica study. What we identified is that genetic variants in these Wnt signaling genes were associated with lung function in both the Childhood Asthma Management Program as well as the Costa Rica cohort. And as you can see in Table 2 on this slide, that there were replicable associations with lung function What this tells us is that these associations are less likely due to chance alone. So what do these preliminary data tell us? They tell us that gene expression in human lung development can really help us identify some novel biology that's independent of histologic stage and can add to the armamentarium of methods to really help us understand underlying lung developmental biology. In addition, we've identified a variety of lung development genes that are really important in normal human lung development, and that lung development genes really harbor these genetic polymorphisms that impact abnormal lung function in asthmatic populations. The question that arises from this preliminary data and based on these observations is really, what is the role of these genetic variants in terms of its impact on intrauterine gene expression? And when we look at genetic variants, they're really important if we can identify some of the functional impact of these genetic variants during lung development. And so we next look to see if we could identify genetic variants that influence intrauterine gene expression in human fetal lung tissue. To briefly give you an idea of why we took this approach is if you look at genome-wide association data in asthma susceptibility, there have been a variety of different loci that have been associated with asthma susceptibility, including ORMDL3, which is one of the most replicated associations with childhood asthma. In addition, Blanca Himes, when she was at the Channing laboratory, identified variants in the PDE4D gene that were associated with childhood asthma in the childhood asthma management program. What's notable about the use of genetic association data using genome-wide genotypic information is the fact that if you look at each of this is a Manhattan plot that shows these genetic associations. Now, for those of you who aren't as familiar with genetic association information, what you can see here is that each of these dots represents a genetic loci. And what we do is we do a statistical test to determine association with these particular genetic loci with a phenotype of interest. In this case, it was asthma. What you can see is that after a statistical correction for each of the tests performed, which there are thousands over the entire human genome, very few of the genetic associations actually pass muster after statistical correction. But what's important is that if you look at these other genetic variants that don't meet statistical correction, there's a lot of really important biology that can be identified here if we can really look at the functional impact of some of this genetic variation. And so although there are a variety of different methods to kind of bore down on this additional biology, the method that we took is to actually look at the functional impact of these genetic variation by taking a look to see if these genetic variants impacted gene expression in human fetal lung tissue. So we hypothesized, again, that these important lung development genes that we know to be expressed in the airway branching time frame of lung development have genetic polymorphisms that are associated with intrauterine fetal lung gene expression, and that these genetic variants subsequently confer increased asthma susceptibility risk later in life. And the approach we took to looking at this is called expression quantitative trait loci mapping, or eqtl mapping. It's very simplistically, what eqtl mapping is these are genetic studies aimed at identifying sequence variants that confer differences in levels of gene expression. And simplistically, you can see this idea or this concept illustrated here. So take, for example, a single genetic variant, a site, at which you can have an A or a T allele. If an individual has an A allele, it allows for the transcription factor to bind to the promoter site very nicely and efficiently, and actually increases the expression of that particular gene. However, if an individual has a T allele at that particular site, the transcription factor does not bind effectively, and you get reduced expression of the gene. So for that particular polymorphism, if an individual has an AA genotype, it's associated with high levels of expression of the gene. And if an individual has a TT genotype at that locus, it's associated with low levels of expression. So the importance of using this methodology is we can identify sequence variants that are associated and impact gene expression. What we did is we did genome-wide eqtl mapping using 300 human fetal lung tissue samples with genome-wide genotypic data and genome-wide gene expression data. The methods you can see here, and I will tell you, I'm not going to go into detail here, but we looked at cis-acting eqtls. These are genetic variants that are within 50 kb of the transcription start site to look at to see if they were associated with gene expression in human fetal lung tissue. What you see here are the results of our eqtl analysis. And this is, again, a Manhattan plot looking at the genetic variants that are influential in terms of intrauterine gene expression. And what we identified was 87,000 eqtls that were genome-wide significant at a Bonferroni adjusted p-value. What this tells us is, while this appears to be more than what we would expect in normal eqtl analyses, what it tells us is that lung development is an incredibly complex regulated process, and eqtls, which are genetic variants that influence gene expression, are really important in the early parts of lung development. This, for example, is one of the eqtls that we identified, which is PPIL3. And as you can see, the different genotypes confer differences in gene expression. So a nice illustration of a fetal lung eqtl. This particular gene is on chromosome 1 and impacts mRNA splicing. We went on to look at the genes that were actually regulated by these eqtls, and what we identified are some really interesting biologic pathways. And these include metabolic pathways like glycolysis and glycolytic pathways, things like adipogenesis and lipid biology, and then fatty acid metabolism. Furthermore, we identified some role of ciliary function, which is also the first time point in which ciliated genes and ciliated cells are notable in airway development. Now, this is really associative, right? Showing us that there's associations of genes that are regulated by genetic polymorphisms, but really is the first indicator that metabolism and metabolic pathways are really important in early airway development, and shows us the potential for environmental perturbations in the intrauterine time period actually impacting metabolic regulation during lung development. We went on to look at the eqtls and look to see, are these eqtls associated with asthma susceptibility and susceptibility to other airways diseases? Furthermore, we looked to see if these eqtls were enriched in genome-wide hits for lung function impairment. And what you can see is that our fetal lung eqtls were enriched for overlapping eqtls in asthma susceptibility, in COPD and emphysema, in interstitial lung diseases, and as well as lung function. And just as note, we looked to see if we could identify these eqtls in other diseases that were not related to respiratory disease, like, for example, macular degeneration, and these eqtls were not linked to that. So what we next did is we looked at these eqtls and we looked at the amount of expression variation during lung development and how it was, how these eqtls were able, how much of the gene expression variation were able to be explained by eqtls. And this plot actually shows the percentage of variation explained by these different characteristics. As you can see, a large proportion of the genes that are expressed in early lung development are explained by eqtls. So if you look at a particular gene that's expressed in the human genome, eqtls explain roughly 1 to 68% of the gene expression variation. In total, if you look at the overall genes, the median amount of gene expression variation explained is about 6% of the gene expression is actually explained by eqtls. What does that tell us? What that tells us is eqtls are important in terms of regulation of gene expression during lung development, but they don't explain the entire complexity of gene expression variation in lung development. And it really argues for additional mechanisms of gene expression regulation that need to be investigated to really understand fully the gene expression variation that we see during normal human lung development. So what we next did is we took a look at these eqtls and see where they sit in regard to the human genome. And what we identified by looking at epigenetic elements in a fetal lung cell line, that these eqtls really overlapped with some very important regulators of of the genome, which include DNA's hypersensitivity sites as well as histone modification sites. And so what this argues for is investigation of epigenetic markers and regulators of gene expression during lung development. So if we look at the EQTL analysis, what it really tells us is that gene expression is under genetic control to a large degree in human fetal lung development, and they impact a myriad of metabolic pathways and ciliary function in early lung development. Again, giving us a clue that metabolic regulation may be really important in terms of lung development and airway biology. Furthermore, genetic variants that influence fetal lung gene expression are more likely to be associated with postnatal respiratory disease, and this has been shown in other previous existing literature on EQTLs. Finally, lung development EQTLs are more likely to be in regulatory elements, and again, gives us some additional evidence to support looking at epigenetic regulation of gene expression during human lung development. And in order to do that, we looked at microRNAs. Now, microRNAs are one epigenetic form that actually influences regulation of gene expression. And microRNAs are a family of endogenous small non-coding RNA molecules. They really modulate physiologic and pathological processes by post-transcriptional regulation of gene expression by binding to target mRNA. And because a given microRNA can impact a variety of different genes, so from hundreds of genes to thousands of genes, these molecules are a really important regulator of underlying molecular biology and can really fine-tune processes, including development. So we did microRNA sequencing on the 300 human fetal lung tissue samples that I've already previously shown you. And what we did is look at gene expression, microRNA expression variation during the fetal lung time periods. And what you can see here is a plot again of principal components looking at gene expression variation, where you can very nicely see that age is incorporated or changes expression of these microRNAs. I think it's important to recognize that microRNA expression changes in a variety of different patterns. For example, some microRNAs can increase, others decrease. Many have nonlinear patterns to gene expression during airway development. This figure is a heat map simply to show that if you look at the top age-associated microRNAs, there are variability of gene expression or microRNA expression patterns that exist in these airway developmental timeframes. Importantly, one of the most differentially expressed microRNAs, which is microRNA 483, which you can see here, is a microRNA that very tightly regulates insulin-like growth factor 1, or IGF-1, which is also important in metabolic control. It's important in organogenesis and really impactful in terms of metabolic regulation. What we went on to do is look at network biology to identify how these microRNAs are impacting gene expression. And using SMCCNet, we looked at a microRNA-mRNA correlation module. And what you can see here is a very complicated figure that shows the interactions of age-specific microRNAs with gene expression during human fetal lung development. If you look at these genes and these microRNAs and pathway enrichment, what you will see is that these genes and these microRNAs are really important in terms of airway development and airway biology. And very interestingly, if you look at this particular microRNA, which is 195-5P, which really is a microRNA that is one of the hubs for this particular network, it has been shown to be impactful in terms of metabolic regulation in myocardial tissue in patients with heart failure. It actually influences the SIRT3 gene that results in some changes in energy metabolism in the human myocardium. Again, giving some credence to the importance of metabolic regulation during early lung development. So these microRNAs are important. We know they're key regulators of human lung development, and we can show, based on human fetal lung tissue, that they regulate these complex gene expression processes during airway development. Furthermore, they impact a variety of metabolic functions that may result in some asthma susceptibility later in life. And microRNAs, again, are really important regulators of lung gene expression and are impactful in terms of their regulation of metabolic features in the intrauterine time period. So now we've spent the last several minutes really talking about what happens in the context of normal lung development. So the question really is, if you were to introduce an environmental perturbation in the context of the intrauterine environment, how does it change normal lung development? And does that have the potential to lead to susceptibility to disease, in particular asthma, as it is an airways disease? So when we think about normal lung development, what we can conclude is that genomics can identify metabolic pathways. These are things like fatty acid metabolism, glycolysis, IGF signaling that are active during early airway development. And that genetic variants that influence fetal lung gene expression are enriched for genes involved in metabolomic features or metabolism overall. And that microRNAs that are most highly differentially expressed during the pseudoglandular stage also change IGF signaling, which may also impair other metabolic pathways. So in total, if we look at the kind of features of normal lung development, it really demonstrates the importance of tight metabolic regulation during branching morphogenesis. And again, looking at these metabolic pathways in the context of intrauterine perturbations may be really important in terms of identifying some of the biologic basis of disease. So the way we looked at a very common, important intrauterine perturbation, environmental perturbation, is we looked at intrauterine smoke exposure. Now, in spite of all we know about smoking and its bad health impacts, approximately 14% of women continue to smoke during pregnancy. And if you look at state-specific rates of smoking, in places like the South, for example, there can be state-specific rates as high as 40 to 45% of pregnant women who continue to smoke. Now, while we recognize that smoking cessation programs do have moved the needle in terms of trying to get women to stop smoking prior to pregnancy, what we recognize is that since women continue to smoke, we really need to understand what happens to fetal lung programming so we can identify potential ways to mitigate the impact on the fetus. So unfortunately, intrauterine smoke exposure remains really among the most common preventable causes of infant morbidity and mortality. And if you look at the epidemiologic data, intrauterine smoke exposure is associated with early onset wheeze, lower lung function, and subsequently asthma susceptibility. If we think about the health outcomes associated with intrauterine smoke exposure, they're both short-term and long-term impacts. For example, intrauterine smoke exposure is associated with preterm both in human subjects as well as in mouse models. It's been shown in multiple studies to be associated with fetal growth restriction and low birth weight, and then reduced alveolarization in animal models. In the long-term risk of intrauterine smoke exposure include an increased risk of being overweight or obese in the neonate and postnatal period. Furthermore, it's associated with increased wheeze and asthma susceptibility. And finally, it's associated with impaired lung function and airway hyperresponsiveness. The way we looked at intrauterine smoke exposure is really to look at nicotine and nicotine metabolism in the fetus. Now, what you can see here is nicotine actually enters the fetal circulation via the placenta, and notably and importantly, there is no evidence of nicotine metabolism in the placental tissues. So if you're able to look at nicotine and nicotine metabolites in the placenta, you can infer what the smoke exposure status is of the fetus. So in collaboration with Dr. Leder's group at Children's Mercy Hospital in Kansas City, we measured cotinine, which is a well-known nicotine metabolite in the placental tissues of each of the concurrent human fetal lung tissue samples. What we identified is that placental cotinine values of greater than 7.5 nanograms per gram tissue identified with high sensitivity and high specificity these fetal tissues as being smoke exposed or not smoke exposed. And importantly, this cutoff showed that it could induce a change in xenobiotic metabolizing enzymes, including CYP1B1, which is a very well-known gene to be dysregulated by smoke exposure. So we characterized these human fetal lung tissue samples as having placental cotinine values of 0, so no exposure, versus those that had intrauterine smoke exposure had placental cotinine values of around 60 nanograms per gram tissue, so a high level of nicotine exposure. What we initially started with was differential expression to identify genes that were differentially expressed by intrauterine smoke exposure. And what we identified is roughly 5,000 genes that were differentially expressed, including microRNAs, the CYP1B1 gene, which I've already previously discussed, and then some really important genes in terms of airway remodeling, TGF-beta signaling, for example, and then genes associated with extracellular matrix deposition. Importantly, as you can see here, this is just one of the differentially expressed genes, again showing that this cut point of intrauterine smoke exposure at 7.5 nanograms per gram tissue really reflects a change in gene expression that would be expected due to smoke exposure alone. When we looked at the genes, the 5,000 genes that were differentially expressed by intrauterine smoke exposure, the pathways that were dysregulated by smoke exposure are, again, really important, interesting metabolic pathways. Notably, it changes genes involved in organ development, which one would expect in the context of normal lung development. But it also impacts a variety of other metabolic pathways, including adipogenesis, fatty acid metabolism, and again, ciliary function. Finally, importantly, it also regulates a variety of different cancer cell types and cancer pathways, suggesting that this intrauterine smoke exposure may be the first hit in terms of the two-hit model for the development of postnatal cancers. So it's particularly concerning to see that these features are already regulated in early lung development. So again, we note the importance of these metabolic derangements that occur after exposure to intrauterine smoke. We next looked at differential correlation analysis, and very simplistically what this method does is it looks to see sets of genes that are correlated amongst individuals with the same exposure, but are differentially correlated between different exposure types. And if you look at the highest correlated modules, what you will see, again, is that there are some very complex regulation of some metabolic features, including glucose metabolism, interferon signaling, which is important in asthma biology, and finally, ciliary function and movement, suggesting, again, that if you look at what happens to the human fetal lung related to intrauterine smoke exposure, is that what we can see is that it really impacts the potential for metabolic dysregulation during airway development, and that might be a key feature to why individuals go on to develop increased airway inflammation and derangements in lung function longer term. Why this is important is this information that we've identified for the first time in human fetal lung has been corroborated in animal models. So now this busy slide is actually beautiful work from Demel and colleagues that used a murine model of intrauterine smoke. And the reason I show this important network is because if you look at the master regulator of this intrauterine smoke exposure network, it is IGF-1, which is insulin-like growth factor 1. Why is this important? It's because this is a key gene of lung development. It's dysregulated by intrauterine smoke exposure, both what now that we've shown in our human data, but now in the mouse model that confirms this analysis. And furthermore, it's really impactful in terms of these metabolic pathways that we've seen throughout the context of normal lung development, and now we're showing is deranged in the context of this intrauterine exposure. If you look at IGF-1 in the same mouse model, it also has some other interesting features that I'll highlight here. So if you look at IGF-1, it's increased in its expression in the mice that have been smoke exposed. And so these are the maternal, the pregnant mice. What you will see is that the maternal mice that had smoking exposure had lower lung weight overall. And the fetus and the neonate of the mouse that was smoke exposed had lower birth weight, had derangements in lung function or lower lung function based on physiologic measurements after after intrauterine exposure. And interestingly, these mice often go on to develop central adiposity later in life. So again, suggesting that potentially some of these mechanisms are resulting in metabolic derangements that have long-lasting implications. Now, these are all associative and not causal in nature, but these are things that actually influence kind of our next set of hypotheses for further investigation in the next few years. So what we now hypothesize is this really nice model of metabolic dysregulation, but where it's been really nicely studied in the context of obese asthma, what we suspect is that these metabolic dysregulation is actually initially happening in the intrauterine context and time frame. So this is data from Baffy and colleagues where you can see that there are a variety of different metabolic derangements, including the development of metabolic syndrome and asthmatics that have a variety of implications in terms of airway inflammation, airways hyperresponsiveness, and subepithelial fibrosis, which are all key features of lung function derangements in subsets of individuals with asthma. So as you can see here, there's dyslipidemia and fatty acid-induced inflammation. There's insulin resistance and hyperglycemia that impacts a variety of different, not only fetal lung maturation, but a variety of other different pathways that influence airflow obstruction. You can have abdominal adiposity as well as obesity that impacts, again, insulin like growth factor 1, and really can result in a variety of different, again, mechanisms that are associated with impaired lung function, things like subepithelial fibrosis, airway, smooth muscle hyperplasia, and bronchial hyperresponsiveness. So in total, if we look at this metabolic dysregulation, if this is really happening in the context of in the intrauterine time frame, it may be the first setup to dysregulated metabolism in the airway postnatally that results in lung function decrements over time. Why is this important? Because if you look at a variety of epidemiologic data, metabolic syndrome has been associated with not only asthma susceptibility, but impaired lung function. And this is really nicely illustrated here. This was a cross-sectional study of 1,000 patients from Paris, where they identified that abdominal adiposity was one of the largest predictors of lung function decrements, including decreases in FEV1 and FVC. But notably, both glucose management and hyperlipidemia were also risk factors for reduced lung function. Again, tying in this idea that metabolic derangements can actually influence airway inflammation and impaired lung function overall. So when I think about kind of the next steps in terms of how we're actually going to kind of disentangle what's happening, is now we have associative data suggesting that metabolic dysregulation in the early time periods of lung development have the potential to impact fetal lung airway development, postnatal airway development. But we also have shown that intrauterine smoke exposure impacts these metabolic pathways and may influence disease susceptibility later in life. But the way to kind of understand the causal link between these pathways is to really investigate these metabolic dysregulation in a murine model of intrauterine smoke exposure. And so that is work that we will be currently undertaking to see if we can actually identify a causal link between the intrauterine metabolic disarrangements with airflow obstruction in the postnatal period. In addition, in collaboration with my colleague and friend, Fernando Holguin, at the University of Colorado, we will be investigating these pathways in the obese, non-atopic asthmatics using gene expression signatures based on endobronchial brushes in obese asthmatics, and comparing these individuals who have metabolic syndrome with those who do not, so we can see if these pathways are dysregulated by kind of the feature of metabolic syndrome. And finally, we will be able to use a variety of additional omics technologies to see if we can create kind of this master integrative network of lung development that incorporates additional genomic data types. And this, I think, really has the hope of being the holy grail to some degree for what we are doing in the context of lung development. If we can model additional environmental exposures, things like house dust mite, for example, proximity to roadway, ozone, air pollution, We can use phenotypic parameters in terms of airway, size, and CT findings. Finally, using genetics and genomics and other features of omics, including metabolomics and single-cell RNA sequencing, we hope to create a master network of lung development that may further drill down on a single pathway, including a single metabolic pathway for additional investigation in this timeframe of lung development. So what I hope I've done over the last 45 minutes is really show you that integrative genomic methods has really identified important metabolic pathways. These are things like fatty acid metabolism, glycolysis, and IGF signaling that are active during human lung development and may impact subsequent diseases like asthma. Furthermore, things like intrauterine smoke exposure. Now, this is just a model environmental perturbation in the intrauterine context, but it really modifies IGF signaling and potentially impacts ongoing metabolic dysregulation that has been shown to result in subsequent lung function decline in epidemiologic studies. Furthermore, additional investigations into the impact of intrauterine metabolic derangements may really be helpful to further elucidate the biologic mechanisms that underlie abnormal lung growth and the development of subsequent disease like asthma. And more broadly, I hope that I've shown you that genomic investigation in human fetal lung development has really identified genes that influence lung function and asthma susceptibility later in life, and that using integrative genomic approaches in human populations can really be used to identify some novel biology and novel susceptibility loci. We've identified numerous cis-eqtls in human lung development that have been enriched for genome-wide genotypic hits for diseases like asthma and lung function, and we demonstrate the developmental origins of asthma using genomic applications. So I would like to just very briefly thank the group at my research group at the University of Colorado, including Dr. Fernando Holguin, who is not only a great friend, but a mentor and has been so helpful in terms of my career. I would like to highlight the work of both Chu Lu and Dr. Katerina Kekris, who have been instrumental in doing the data analyses that I've shown today, and our incredible group who you can see here who make it really a pleasure to come to work every day. So thank you to that group. In addition, I would like to thank the incredible group of collaborators at the Channing Division of Network Medicine, including Scott Weiss, Benji Raby, who is also at Children's Hospital, Kellen Tanasira, who is now at UCSD. I'd like to highlight Alvin Koh, who has been an incredible collaborator on this project over the last several years, Dr. Vincent Carey, who taught me about genomics, Les Kobzik, who is just a giant in developmental biology, and a special thanks to Kathleen Haley, who's working in the MICU today, but who has been so important in this work, and finally, Dr. Don DeMeo, who's been instrumental in the Fetal Lung Project. And finally, another special thanks to the group at Children's Mercy Hospital who really gave us access to this tissue, who came up with a lot of the scientific inquiry regarding lung developmental biology, and who have been incredibly influential in terms of my career. And finally, this funding, which I am eternally grateful for. Thank you very much, and thank you, Chris, for the opportunity. I will stop there and take questions.
Oh, wonderful. Let me see if we have some questions online. Sunita, thank you so much. It's terrific. I'd love great science, beautifully presented. What could make one happier? And your ability to bring in so many different aspects of lung development and asthma, it's terrific. Thank you. Can I just begin by asking, I was curious about the 300 fetal samples that you use for analysis. Do we know anything? Are they all healthy, normal origins or disease-prone, or what do we know about those samples?
Yeah, that's an important question, and I apologize. I should have addressed that up front. So if you look at these human fetal lung tissue samples, these will refer from spontaneously and electively aborted fetal tissues. All we can say about these tissues is that we have access to the fetal foot size, which gives you an approximated estimated gestational age. And then we have the identified sex of the fetal tissue. Notably, we don't have any additional information about the mom or the fetus, but what we are able to infer is based on gene expression. So we can do outlier, we removed outliers from the analysis, which is why some of these, you can see some of these data weren't necessarily for the 300 samples. But we were able to actually take out any of the samples that had genotypic abnormalities or had gene expression abnormalities, and we confirmed the sex based on methylation, and that's work that was done by Don DeMeo's group.
Nora Barrett has sent you hello and congratulations and great talk, and asks, are there measurable changes in fetal metabolism in moms who are on therapeutics such as metformin and other drugs that can be helpful for analysis? And I wondered the similar about, what about diabetics and the impact on gene expression in fetal development?
Yes.
Similar.
Oh, that's a good. So first I'll start by saying, hi, Nora. Thank you for the question. Yeah, this is an incredibly important area of investigation. We can infer some information based on kind of mouse models that demonstrate that there are changes in gene expression in fetal lung, in the fetal lung tissue of a mouse model based on glucose dysregulation during pregnancy. We don't have we don't have human data to corroborate those findings yet. What I can tell you, however, is that if you look at glucose dysregulation during pregnancy, it has been associated with changes in IGF expression levels in the cord blood, as well as IGF levels in the peripheral blood PBM or the PBMCs in the postnatal period. So it really suggests that glucose metabolism and glucose changes in glucose during lung development, and it's important. And notably, one thing that has been shown in physiologic measurements is that if you look at hyperglycemia during pregnancy, it actually increases the fetus's the respiration, the number of respiration events that occur in the fetus. And why that's important is because actual respiration is important in airway development, lung development. And so one of the suppositions is that by changing glucose metabolism in the mom or during pregnancy, it can really impact kind of lung morphology based on respiration and its changes there. But that impact is undoubtedly related to changes in gene expression. It just hasn't been shown in the human context yet.
Your friends and admirers are checking in.
Oh.
And Nancy Lang says hi and wants to ask about associations for your gene findings in not just lung growth and development, lung function, but are there similar associations with immune function genes, specifically as might be related to atopy?
Yeah, that's a great question. Hi, Nancy. Oh, it's so good to hear from all of you. So thank you for being here today. I will also say that. So thank you. Yeah, that's a great question. So I didn't actually focus on immune dysregulation, but I can tell you with certainty that intrauterine smoke exposure impacts the innate immune genes in fetal lung tissue. What I can't tell you is that intrauterine smoke exposure changes the imprint in terms of what happens in eosinophilic-driven disease versus non-eosinophilic disease. That we don't have a signature for. But it does change the innate immune response. And one other intriguing thing based on murine models is that the change in innate immune response that occurs during lung development may have some impact on postnatal susceptibility to infections, things like viruses and bacterial infections that we know then go on to result in susceptibility to diseases like asthma. But I can't tell you that it changes polarization in terms of Th1 versus Th2 inflammation. But what I can tell you is that the immune response appears to be dysregulated in the context of intrauterine smoke.
Great talk, says Elliot Israel, and asks, is there anything to be learned from the phase of saccular differentiation in fetal development?
Yes. Oh, yeah. Hi, Elliot. Yeah, thank you for the great question. There is no doubt that we could learn more because you get terminal bronchial differentiation that occurs during the saccular stage of development. And we could no doubt identify some additional kind of genomic underpinnings of disease if we were able to look at that time frame. The problem with doing that is that getting access to tissues that are later in gestation are more challenging in terms of due to ethical reasons. And so we don't have access to that data. And so what we're limited by is the ability to look at the saccular and alveolar stages of development using murine or other animal models. But there is no doubt there is a lot of important airway biology that occurs there, and there's likely to be some signatures that influence asthma susceptibility during those time frames.
Sunita, do you find any overlap of developmental eqtls in lung and asthma susceptibility with early allergy in other tissues, such as gut and the skin, because these tissues share many key developmental pathways?
Yeah, that's another great question. Yeah, so if you look at development, so many of these organ systems really share developmental origins, and there is likely to be overlap between genetic variants that influence gene expression in the lung with genetic variants that influence, eqtls and these other data types. I will say our group has not looked at that to date, but that is definitely something that would be interesting to look at, because we know that eqtls not only influence asthma susceptibility, but they also predispose to a variety of other autoimmune conditions. And so looking at other tissue types, some of these are likely to be tissue specific, but clearly not all. And so there is no doubt it would be really interesting to be able to look at these different tissues overall, but we have not done that to date, but we will. So thank you for the suggestion.
Maybe I get one last question that I was interested in the challenge that you have in distinguishing developmental impact on lung function and reduced FEV1 or ratio and asthma, and how to distinguish the predisposition one from the other. Will you be able to do that? So, okay, another excellent question. With the human data, it's going to be very hard to disentangle. I think we have to go back to the murine model or a primate model of development to really look at that. And again, a lot of this is really associative in nature based on what I'm showing you today, but it really, I think, creates a hypothesis or idea to really investigate in a murine model so we can actually see, is this just airway remodeling and decrements in lung function, or is this really predisposing to asthma susceptibility? Well, a lot of virtual clapping for a terrific presentation. Thank you all for your questions and participation. And thank you, and we look forward to seeing you next season. Take care. Thanks.